learning or the artificial intelligence methods are used in many areas, and then briefly introduce in very simple terms what these methods are, and then we'll talk about some applications in molecular modeling, right, in the first half of the session. So in the second half of the session, what we will do is we will take some examples of how these methods are used in drug discovery. Okay. So if there are any questions, please feel free to interrupt anytime. You don't have to necessarily wait until the end. So when I'm presenting, I may not be able to see the chat. If if someone can prompt if there are any questions in the chat, maybe like
Okay,
and
sir.
if you have a question, just interrupt me and ask the question also. I have no problem. Yeah. So okay, so as I said, I'm going to talk about the modern machine learning methods or the artificial intelligence methods and how they are applied in molecular sciences or in molecular modeling. Okay, so I will turn off my video and go to full screen. I hope you can continue to see my screen, right? Yeah, you can see my screen. Can you confirm?
Yes, sir.
Okay. All right. So, as it was mentioned, I'm a faculty member at IIIT Hyderabad, and I'm associated with the Center for Computational Natural Sciences and Bioinformatics, and also an innovation hub called iHub Data. Okay, so in the last, I would say about 5 years, right? So we have been seeing so much usage of these terms, right? People talk about artificial intelligence, machine learning, deep learning, and so on, right? So it has become so common, right? So whether in news articles or whether among discussions among people, new papers that come, breakthroughs that happen in technology, and all of that, right? So, we will slowly understand or try to touch upon what one of them at least, right? So then what we also know is that these methods are applied in many, many different areas, right? So some of the common applications are given here, right? So can maybe you can type in the chat or answer. That's okay. Can you give one example of an artificial intelligence method that we use in day-to-day life, right? That we encounter every day almost. Can you give some examples? Yeah, I mean, just take any example that where we are using AI, right? Someone says YouTube, okay? Google, okay? Anything else? Right, okay, let's take YouTube, right? So YouTube is given as an example. So let us assume all of us open our browser now and then go to youtube.com, right? So the appearance or the different kinds of videos, right? All of them are very different, or in some sense, the homepage of YouTube will be very, very different from each one of us, right? It's not like someone is sitting there and then saying, oh, Deva is going to youtube.com, so I have to put these kind of videos in the first page, right? Similarly, Vinay, who has given this example, right? Mr. Vinay Kumar. So, if he goes to YouTube, there's no people or there's no person behind the system who decides which videos to show in the first page, right? But most of the times, if you notice, these videos are relevant to us in the sense that the system has the history of our behavior, in the sense that it knows what are the kind of videos that we have watched. Even among the videos that we have watched, which parts of the videos we have, for example, watched again, or sometimes we have skipped the video and things like that, right? So there is this history which is getting used to display the kind of videos that we may be interested in. And most of the times, and this is good for us in the sense that these are the videos that we want to watch possibly also, right? So depending on each of our interests. Similarly, if you take social media like Facebook or Twitter and things like that, right? So a lot of times it shows, do you want to be friends with this person, or do you want to follow this person on Twitter, right? Again, most of the times, these are people who we may already know, or who we may be loosely connected to. Sometimes we ourselves are surprised that, actually Facebook found somebody who the Facebook thinks that is known to us. We are not actually looking for that person on Facebook, but somehow it appears and we are happy that we found this person and we could connect with them, right? So similarly, you have lots of other examples where, for example, if you go buy stuff online, right? Each of our homepage is different with products that are much more relevant to personalized in some sense, right? And similarly, like flight tickets, the costing of flight tickets. We know that the cost of the flight tickets change every day, right? These are very dynamic. It's not like, again, a group of people sitting and deciding what should the cost be, right? So maybe there are some rules, but in addition to that, there are AI algorithms that maximize the profit for these companies, right? And we can come up with many such examples, right? Similarly, in healthcare, we have many systems now, like you have robotic surgeries, for example, or we have many systems now that can make things faster. For example, if you do biopsy or a pathology, right? The time it takes to possibly identify cancerous cells in a whole slide image, it takes a long time, right? But then you can have artificial intelligence methods that can quickly process these very large images and then give us decisions which are as close to an expert pathologist, for example, right? Okay, so these are some of the examples, and we find these methods, in our day-to-day life, almost everywhere these days, right? And of course, recently, this ChatGPT which came, which took everybody by surprise, and a lot of us use that also, right? Okay, so how does this? What are we talking about today? What we are talking about today is to see how these artificial intelligence methods can be applied in drug discovery or molecular modeling. Right? That's what our interest is, right? Not shopping or social media or anything else, but we are interested in finding out how these AI methods are used in molecular modeling, right? Okay, so when you talk about fundamental sciences, let's say molecular modeling, right? So these three are the three main paradigms that we have been using for making new discoveries, or or furthering or advancing the science, right? So first is the experiments, right? So all the discoveries start with experiments and end with experiments, most of them at least, right? So what does it mean? Experiments are the ones which will lead to real life applications, right? But what is the role of theory? For example, if you have a set of experiments and you make some observations, you can formulate some theory, and then you can try and understand why you made certain observations, right? So that's what we make observations in experiments, and then we use, we formulate theories to understand these experiments. So once this, there is a reasonable understanding of the experimental observations via theoretical principles, right? And then we accept the theory saying, oh, this theory seems to explain most of the observations that we made, and hence we expect, hence we accept this as a certain, a solid theory or a solid theory, right? So once the theory is well formulated and is accepted, then we can do computations in the sense that we can use this theory and then convert or code it into a program and then start making predictions. Okay, so we make predictions. So we don't have to, at this point, we don't have to necessarily do all the experiments to, come up with new discoveries, right? Because experiments in general are very expensive, and then they take a lot of time. And then we also consume a lot of resources in experiments, which is not very ideal. Or someone says voice not audible. Can you? No one can hear me. I did not do anything, and it was just how it was before.
Sir, your voice was audible, sir.
It is okay.
Okay, sir.
Okay, so I think
Vinay Kumar Garu, please check your connections.
okay, so and we make predictions, and when we make predictions, we can do better experiments, or we can reduce the number of experiments that we have to do in order for coming up with real life applications, right? So this is the three paradigms in which we have been, in some sense, the scientific research works, right? So, experiments, theory, and computations. So now, what has happened in the last few years is that, or over the century or more, we have generated large amount of data. What is it? What do you mean by data? When we do experiments, we make observations, right? So that is a data point. So similarly, when we do a computation, we predict something. That is a data point. Now the question is it possible to use the data coming out of experiments or data coming from computations to come up with new insights? Or is there a better way or a new way of predicting properties of molecules, for example, right? So that's basically what is called the machine learning, right? Where we use the existing data and then use that data to understand, right? So once, similar to theory, similar in some sense, very, very different in other sense, right? Similar in the sense that we try to come to some kind of understanding based on the experimental data. It is different in the sense that, the understanding here is that of the machine, and here the understanding is that of an expert or a human being, right? Okay, someone else also says it's not showing video and no audio. So I will continue while you can worry about it. So, the question is, can we use machine learning to understand the data? From there, you start doing predictions, right? So now you have, two ways of doing predictions. One is the traditional way where you use the theory to predict, and then the second one is to use the existing data and come up with machine learning algorithms and start predicting newer things. Okay, so we will come back to this later. But how does the typical computations work? The typical computation work in the sense that we have theory, and that is we write a code to deploy or implement the theory, right? Let's talk in terms of molecules. Okay, so let us assume I am interested in predicting molecular properties, right? Let's say bioactivity of a molecule or toxicity of a molecule, things like that, right? So for a given molecule, which is the input, right, I want to, calculate the toxicity of that particular molecule or predict the toxicity of that molecule. So typically what we have is we have a certain theory, and this is not a great example because the theory of toxicity is not very well done, but so let us assume binding affinity, right? For a given protein and ligand combination, I want to calculate the binding affinity. right? So for example, we could use statistical mechanics, classical mechanics, and do code, use codes like GROMACS or NAMD or CHARM and things like that, and use a computer. And with respect to a protein and a ligand, we can calculate binding affinity. Okay, so that's what we normally do. On the other hand, if we take machine learning, right? So what we do is we swap this output and the code, in the sense that we give the input and the output. How do we get the input and output? This is coming from the historical data, right? That's what we spoke about, right? So we use the existing data. Existing data mean for a set of protein ligand complexes, for example, I know the binding affinity, right, from the literature, right? So I have the protein ligand complexes here. I have the binding affinity here. Now I ask the computer to figure out a code or figure out a program that will take the input and gives a certain output. That's basically what I do. So once I can figure this out, I can do a typical computation where for a future input molecule, for a new protein ligand complex, I can use this code generated by a computer and then use a computer to get the outputs. That's a fundamental difference between the typical computations and machine learning. Okay, so let's make it slightly simple. So today, right, what we are saying is for a given molecule, let us say I want to calculate the energy of the molecule. Okay, so what we could do, we represent the molecule in a certain way. So most of the times we represent the molecule in terms of the atomic coordinates of all the atoms in the molecule, which is given as Zi, and then Ri is the position of all the nuclei in the system, right? Let's say the Cartesian coordinates. So we give this as the input, for example, to Gaussian program, right? And then we get the energy, right? In other words, to make it even simpler, for a given input X, let's call the whole thing as X, right? And we have a program which we just call the function F, okay? And what we want to predict is the energy Y. So we calculate Y as a function of X. So that's what we normally do, right? In other words, X is known, F is known, right? X is the input. We know exactly what the molecule it is that we are trying to calculate the energy for. And then we know the function where it is the quantum mechanical methods. And then I'm calculating Y as a function of X. In machine learning, what we do, let's forget about this X prime for now. So what we do is we have this X which is known and Y which is also known coming from the historical data or the data that already exists, right? Y and X are known. Now I do not know F. Okay, so that is the machine learning algorithm where for the combination of X and Y pairs, you try to come up with some algorithm or a model that will take X as an input and gives Y as the output, right? So we train this model. Let's assume this is a neural network. So we train this neural network such that, for each value of X that is in the data, it takes and does a mathematical operation and gives Y as the output, and it matches whatever data that we have, right? So in other words, X is known, Y is known initially, right? And we model the F part using machine learning algorithms. Okay, this we will skip for now, right? So what did we do so far, right? So what we said was that we have three fundamental paradigms in scientific discovery or the way of in research methodology. One is what are called the experiments, right? So where we do experiments, and second one is the theory or the theoretical, laws, for example, quantum mechanics or thermodynamics or classical mechanics and things like that. And then the third one is the computational science, where we use the theoretical science to do calculations. For example, quantum mechanical calculations or molecular dynamic simulations or docking, for example, right? And things like that. That's the third paradigm. And then the fourth paradigm, which is a recent phenomenon, which is proving to be very, to have very high potential, right? For accelerating science is the data-driven science, or the science which is done using the historical data. Okay. And you can see, I mean, you can really see the explosion of the number of people who are getting into this area of machine learning applied to molecular sciences. Yeah. So this is, for example, is the number of publications not really updated until 2020, that are based in machine learning, right? It's kind of exponentially increasing, right? So, these methods, the AI, ML, and DL methods, again, are used for, different length scales. You can, use these methods to, model things that happen at the atomic or subatomic scale, right? Like, for example, there's a lot of work that goes on in high energy physics, right, where they use the data and try to, identify new anomalies and new phenomena and things like that. And you can do at the small molecular level, for the large molecules like proteins, DNA, and, for example, the whole cell and the organ level and the organism level, right? At different length scale, people have been able to, use these methods reasonably successfully. Okay. And again, these methods are not so new, in the sense that the first paper, if you're able to see, the first paper using some kind of AI method, or the machine learning method, came out 50 years ago, more than 5 decades ago, right? So where they used a simple perceptron to, to classify these molecules called 1,3-dioxanes based on their pharmacological activity, right? Okay, so, and this is what we saw so far, right? So now what we will do, we will try and talk about some problems, or how, right? So in this slide, for example, we saw the 4 paradigms in which we do molecular discovery, right? When do I use? If I already have the theory, and if I'm able to do predictions, then why do I need machine learning, right? So we will give examples of problems where machine learning proves to be, more efficient or more useful than the theoretical method-based computations. Okay. So what we will talk about in the next 20-25 minutes is the different kinds of problems that could be tackled using machine learning, where where it is better than the traditional way of doing computations, right? So the type 1 problem that I will give example, I'll at least give one example each. One is like limited by resources, right? So where the time that the computation takes is very high. In that case, maybe I can use machine learning to make the method more efficient, or make the prediction more efficient. Second one is the theory is not really well established, right? We still don't understand. And in this case, can we use machine learning, right? And then the third problem is we understand things very well, but it's not very straightforward to do computations. In that case, can we do machine learning? And then the fourth one is like new ways of new kind of questions that we can ask, novel problem statements that can be tackled using machine learning. Okay, so let's just take examples, for each of them. Yeah. So first one is, let's say, molecular quantum mechanics or density function theory. right? So the molecular quantum mechanics or the quantum mechanical methods or the density functional theory methods are extremely useful for modeling molecules and predicting the properties, predicting reactions, predicting different kinds of phenomena, and hence very useful in modeling new drugs, new materials, and come up with new hypotheses, right? And they are very helpful. So how do we normally do? So I have a few slides on how we normally do, but I'm gonna skip a few of them. But and maybe let's use this slide. Okay, so in quantum mechanics, what we do is we consider the molecules as boxes filled with particles, which are nuclei and electrons. And the electrons form standing waves in these boxes, and those standing waves can be represented by a function, which is the wave function, right? Schrodinger's postulate says that the wave function contains all the information about that particular molecule if you are able to get it. It also gives a formulation or an equation which helps us get the function, right? So what are we saying? If we can find any given system, whether it's atoms or molecules, all of them are associated with the wave function, right? And if we can get the wave function, we have all the information about the molecule. And how do we get that function? By solving this particular equation, right? So, some of you may be aware of the quantum mechanics possibly. And, this one seems so simple, but when you expand the Hamiltonian of the H here, even for simpler molecules, these equations become extremely complicated. These are second order differential equations, and it's not possible to get analytical solutions for any system beyond, hydrogen atom or hydrogen-like atoms. Okay, so and hence we use quite a approximations to get the energies, but still, I'm going to skip a few slides here. Yeah. So, as I said, we have different kinds of methods, right? So, starting from Hartree-Fock to MP2 and couple cluster and so on, right? So we can use these methods to calculate molecular properties reasonably accurately. We also have methods called semi-empirical methods, which are not so accurate, right? But then, it is useful for modeling larger molecules, very large molecules. And what is more popular these days is the density functional theory methods, right? So, these are very useful. But the problem is these are time consuming, right? So the first limitation is that even for a molecule with few atoms, if you want to use very high level of theory, right, you need to spend a lot of computational time. Yeah, so it's not easy. So, if you want to handle large molecules, you have to use severe approximations. Then that makes the results not so accurate, right? And hence these methods are limited to small molecules. Yeah. And then the next problem is, we all know how important the dynamics of molecules are, right? For example, life exists because of molecules, the dynamics of molecules. Things are not stationary, right? The things are constantly moving, molecules are constantly moving, and then they interact with other molecules, ions, and so on, right? All these are important for the existence of life. If you want to do computations to understand molecular system, considering the dynamics is important. And that is very, very difficult using the quantum mechanical methods, right? And, when it comes to biology, where, for example, I want to model protein ligand interactions or receptor drug interactions, right? So quantum mechanics are, almost out of scope because we just cannot model very, very large systems, right? So this picture covers everything where, this is the accuracy and this is the number of atoms with respect to increasing number of atoms, right? So the accuracy of the quantum mechanical models also go down because we start using severe approximations, or else it's not possible to model, right? And hence what we do is we use molecular mechanics. Molecular mechanics methods are based on potential energy functions, which have very simple equations, right? So we use molecular mechanics methods to model molecules, but then in terms of the accuracy, they are very low. So the question now is it possible to come up with a method, right, which is efficient in terms of the computation, that it is not so difficult to do computations or equivalent to molecular mechanics, and at the same time in terms of accuracy, right, it is almost at least equivalent to quantum mechanics methods, right? So mechanics, worry about it. So the potential energy of the system is given by energy corresponding to bonds, energy corresponding to angles, dihedrals, and then some non-bonds, right? Non-bonded interactions. Yeah. So let's not go into the details. So this is a method where machine learning method where you could use the existing data and then come up with a new model that can predict energies of molecules. Okay, so let us say what I want to do is calculate energies of molecules which have these atoms H, C, O, and N, right? So this is what this slide shows is a simple way of representing these atoms, right? For example, H1 is this one, yeah. So if I want to represent this atom H1, right, I can use 1000, right, which is designated to hydrogen, 0100 for carbon, 0010 for oxygen, and 0001 for nitrogen, yeah. And then the next four vectors, dimensions, that capture the environment. All, for example, if you take this hydrogen and this hydrogen, H1 and H5, they are very, very different, right? For example, this is connected to carbon, but that is connected to this is connected to nitrogen, right? So let's not go into the details, but what I'm trying to say here is that we have to tell the machine learning method what this molecule is, right? So we come up with a certain scheme, and then we represent each bond by a combination of these. This 9 is not correct. This should be 8 plus 8 plus 1. That's 17 dimensions. So you have 8 for atom 1. A bond is formed between two atoms, right? Let's say atom 1 and 2. So that's the 8 characters for atom 1, 8 characters for atom 2, and then that's the distance between the two atoms. So similarly, you can represent the angles, dihedrals, and the non-bonds. Yeah. So now what we do is we start from the Cartesian coordinates of the atoms in the system and the corresponding atomic numbers, right? So we have the atomic numbers Z1, Z2, and so on. And then you have the position of each of these nuclei, R1, R2, or N. So now what we do, this information is good enough to come up with all the bonds, all the angles, all the dihedrals, and all the non-bonds once we know the connectivity information. Okay, so what are we interested in? We are interested in getting the energy of the system. Okay, so we have a neural network which is trained to reproduce the energy that is there in the data, right? So basically that's what is done. Given a set of Cartesian coordinates, we predict the energy. What do we do in between? We have this representation which is done which is generated based on the scheme that I showed in the previous slide. And then you have a neural network here, which has many parameters which can be changed to minimize the energy between, or minimize the energy difference between the predicted value and the actual value. Okay. All right. So just let's look at some examples. You know, what you see in the left is the predicted energy and the ground truth, or the quantum mechanical energy, right? So as you can see, you know, it's a reasonably good prediction, and this shows the distribution of the mean absolute errors, right? And we see it's reasonably well. The mean absolute error is about 1.5 on the test set, which is very, very good, right? We also could test this on molecules which are bigger than in the system. And for example, in this case, we took some random isomers of C11H12, H22, and then get the energies calculated from DFT, right? And then these are the numbers that come from this particular machine learning method. Okay. And then, you know, just for comparison, we also have this semi-empirical method, which is given as this. So what is it we have here? So we can use DFT to calculate the energies. Let us assume it takes about 1 hour to get the energies of these molecules, right? So this particular method takes 1 microsecond, 10⁻⁻⁶ seconds, right? Huge difference. Right? Even assuming this takes 1 second, this is 10⁻⁻⁶ seconds, right? 1,000,000 times faster, right? This is 1,000,000 times faster, but then gives energies which are very, very similar to the quantum mechanical method. Yeah. So that's basically what it is. You can, you know, test other properties like how the energy change with respect to bond stretching and then bond bending, and then we can also calculate reaction energies, for example, with respect to different kinds of reactions. That's the ground truth. This is the predicted values, right? You know, as you can see, these are, you know, very, very good. And then, yeah, this we can skip. And we can also do optimizations. What do you mean by optimizations? You know, we can start from a rough structure of a molecule and then try to get the equilibrium structure, right? So, you know, these are some of the properties, you know, these are just some representative examples, but, you know, these are properties that you can predict in a very, very similar way, right? You can predict a large number of molecular properties like polarizability, dipole moment, and vibrational spectrum, and things like that. And then we can, you know, predict, for example, protein ligand binding affinities, or the ADMET properties of drug-like molecules, protein structures, and things like that. Okay. So this is how you do. Just to summarize, you have a historical data, use some machine learning method to come up with a model which can take an input and then give an output or predict a property. Right. So let's go to the next set of. So far, if there are any questions, please go ahead. I've been continuing. So. Are there any questions? Okay, so let's discuss the next set of examples. Next type of problems, which is limited understanding. What is it we are saying? We're saying that the theory is not very well established, or I still do not know, you know, how to do this or solve this particular problem. Example is, sequence to structure prediction of proteins. Right? Protein, this is basically the protein folding problem, where you have a set of or a sequence of amino acids, right? So coming up with a reliable structure of this molecule is an unsolved problem, right? Using our understanding of the protein folding is very good, but not to the level of predicting structures, right? Using whether it's molecular dynamic simulations or homology modeling or threading methods and so on, right? So we can predict, but then, it's not very accurate, okay? So in this case, one of the examples that, again, many of you may know already is the AlphaFold method, where it takes the protein sequence and predicts the structures very, very accurately, right? So initially it was proposed in 2018, 19, and then a second version came out 2 years later, and it predicts protein structures very, very well, right? So what does it do? It uses the existing protein structures and their sequence, and then comes up with a network or the ML algorithm, which predicts the interatomic distances or inter-residue distances, or the contact map, and the backbone dihedrals. Okay, and from there it it generates multiple models, scores them, and then takes the best models and do minimization to come up with the structure, right? As you can see in this slide. Okay, so the ground truth or the actual structure is given in green, and what is predicted by the AlphaFold method is given in blue, right? As you can see, they are very, very similar, right? So, achieving this level of accuracy is possible using the modern machine learning or the artificial intelligence methods. Okay, and this kind of summarizes how well the AI methods are able to do to accurately predict protein structures. Right. So let's look at the last two problems quickly. One is the not so straightforward problems, and the second one, or new problem predictions. Okay, I'm gonna skip this and talk about that later. So novel predictions, what does it mean? So typically what we do is for a given problem, or sorry, for a given molecule, what we want to predict is the property of that molecule or the properties of that particular molecule, right? So where X is the molecule, Y is the property, right? F is the function which maps X to Y, right? So this is a problem which we solve using typical computations. But the actual question, if you think of it, what we are asking or what we want to solve is to be able to ask this question the other way around. What do you mean by other way around? Where I say that, I want molecules, I want X, which display these particular properties, not the other, not the initial one, where we say, this is the molecule, what is its property, right? And that's a good question. There's no question, there's no issue about it. But what we are saying is, it will be nice if we can solve. I mean, the first, when we use data, right? I mean, if you want to use the data to get the maximum out of it, in the sense that if you want to come up with the best model with a given amount of data, right? So, different kinds of data processing will help in coming up with a better model for sure, right? Because data is common to more, especially the open data, right? Common to everybody. So, but then people come up with different models which are of different accuracy. That is in how you pre-process the data, what kind of features do you use, and of course, this other part is the different kinds of methods or algorithms you can use. So yeah, certainly yes. I mean, there's no prescription on how we could do it. One has to try multiple things to come up with the best model. Okay, so let's continue. In the second part, what I will concentrate more on is how we could use the intelligent systems or the machine learning methods for accelerating drug discovery. Yeah, this is something that we saw already, how the the machine learning or the modern AI methods are coming out to be the fourth paradigm towards scientific discovery. Right. So this is a title. This slide is something that many of you or most of you will be aware of, which is the drug discovery process, where from a given disease, you want to come up with a drug that can be marketed, which will treat that particular disease, right? And we also know how complicated this process is, how it takes a large amount of time, how it is expensive, how many kinds of disciplines that need to contribute towards this process and so on, right? We make this slightly simpler, where we have a disease, right, for which we want to come up with a new drug molecule. Okay, so one of the steps is to identify a target, like a receptor or a protein, most of the times, where this drug will bind to, and intervene or intervene with the cellular processes, which will help in treat the disease. Okay, and then the second one is, for that particular target, we want to come up with a new molecule that will bind. And then once you are convinced, both experimentally, that it actually binds to the target and it works in some animal models, you go for the clinical trials, and if that is successful, you have a marketable drug, right? So in the last few years, if you see, you have number of these methods that appeared. Like, for example, this particular work talks about how machine learning methods can be used to identify the targets for a given disease, right? And this one, for example, talks about once you know what protein you want, right? If you know the sequence, how do you get the structure of the protein? This is AlphaFold paper. And then this paper talks about, once you have this structure of the protein, how can you design, de novo design to identify molecules. Once you identified molecules, how can you synthesize? And this paper talks about, if you have a scheme by which you can do a synthesis, how can you use AI-assisted robots which can do the synthesis for you, right? And all the way up to predicting clinical trial outcomes, right? So the only one of the common aspect of all these works is the integral part of AI methods, right? In all of them, AI or the modern machine learning or deep learning is used to solve these particular problems. And more, people, think there is a lot of potential where each of these tasks in the drug discovery pipeline can be made more efficient using the AI methods, right? So these are some of the tasks that are there that have been already solved using, that people have been trying to solve using AI methods, right? Okay, so let's take some examples. One is, this is related to the absorption and distribution profiles of drug molecules, where we want to predict the solvation free energy of molecules, which is also related to how the solubility, for example, right? So usually what do we do? Yeah, so if I want to get the solubility and predict that using solvation free energy, what we do is, we can do calculations like molecular dynamics calculations, like thermodynamic integration or free energy perturbation methods, right? But these are complex in the sense that these take time, right? For a given solvent and a given drug-like molecule, right? It takes, reasonable computational effort to calculate the solvation free energy. So in this case, what we have used is a machine learning method that can be used to get the solution free energies. Right, so this is how the network is, okay? So I will try to explain to some extent, not large detail, but let's see. So for, let's say I have a drug-like molecule. I hope you can see the cursor. This is a drug-like molecule, right? And this is a solvent molecule, right? For this particular combination, what I want to predict is the solvation free energy, right? I want to predict the solvation free energy. So what we have here is this solute or the drug-like molecule is represented as a graph. Solvent is represented as a graph, right? And then we use a message passing, neural network MPNN to get the features of the drug-like molecule and solvent molecule. That's the solute features and the solvent features, right? So the number of on the columns you have the different features, and the rows you have the different number of, these are the atoms in the solute molecule, right? So similarly here, the atoms in the solvent molecule and the features of each of these atoms and bonds, right? So that's the first phase, which is called the message passing phase, which is, taking the molecules and then getting some features. Now, we combine these features in a certain way. For example, a simple dot product of these two will give us another matrix, which we call the interaction map. And then you combine the solid features, interaction map, and the solvent features in a certain way, and then get all of them together, right? Concatenate to get a long vector, and then use this as an input to a neural network and predict the delta G of solvation. Now you have this huge network where you train this end to end, right? You train this end to end, and then train this model to predict the delta G of solvation. Okay, so now if you look at the results, this is what are the predicted solvation free energies versus the experimental solvation free energies using this method. So as you can see, it's pretty good, right, in terms of predicting the solution free energies. And this particular method, which is Cision, does very well compared to the existing other methods. So the other one we also show is that how this interaction map that I showed here, right, this one is useful in making some chemical interpretations. See, one of the problems with machine learning methods that a lot of us do not like is that these are all black boxes, in the sense that they can predict very well. They can make accurate predictions, but we do not know if they do predictions for the right reasons, or it is actually learning some fundamental concept for the prediction and so on. Okay, so this is something that I showed before, where we have inverse problems, where we said, instead of going from molecules to properties, is it possible to go from properties to molecules? Where you have a method to which you ask that, I want molecules which have a certain set of properties, right? Can the algorithm give us molecules with respect to these queries? So let's think of this pipeline, okay? So where the objective is to synthesize molecules that have specific properties or molecules of interest. Initially, I do not know what these molecules are. The only things I know are I want these molecules to have a certain set of properties, right? So let us assume that I want to synthesize molecules that bind to a certain protein, right? I do not know what those molecules are. But the question is it possible to automate this using AI methods, right? So the first step is to identify molecules with desired properties, right? So basically what we are saying is, if we can solve this inverse problem, where I say I want these properties. Can you give me suggest molecules, right? So that's the inverse problem. Second one is once I have these molecules, let's say that is this one, how do I make these molecules? Predicting the best synthetic route to make those molecules. And then the third one is once I know how to synthesize, can I use automatic methods which can take this scheme and then start making molecules on its own. And then the fourth one is once the synthesis is done and some spectra like NMR spectra or IR spectra and things like that are measured using spectroscopic techniques, can I predict what that molecule is, right? So, I have the spectra, is it possible to tell what that molecule is which gave rise to that spectra? So now the only thing I need to go back is to check this and this. If these two are the same, that means, my objective is done. That, the whole thing is automated in some sense, right? Is it possible to do this? So we'll take example of the first one and then talk about it. Yeah. So let us assume we will talk about three different things. One is I have, let's say, 100 million compounds. I want to choose the top molecules from there based on some criteria, right? And the second method that we'll talk about is how do I then generate new molecules with desired properties. And the third one is, given the NMR spectrum of a molecule, is it possible to identify the molecule, right? So here, for example, let us say this box you have is the known chemical space. What is known chemical space? This is molecules or drug-like molecules that are already known, right? For example, zinc library is something that some of you may be aware of, right? So let's call that as a known chemical space where we know these molecules. That's about 1 billion molecules, 10 power 9 molecules, right? So now my exercise or my task is to identify certain molecules or rank these 1 billion molecules and choose top 1,000, let us say, right? So normally what we should do, and maybe we could do a virtual screening. If I am targeting some protein molecule or receptor molecule, I can do a virtual screening and rank these billion molecules and then get the top molecules. Let us assume these green dots of those molecules of our interest. But then there is a problem. The problem is this is very big, right? I cannot do, for example, docking of 10 power 9 molecules. That is very, very big, right? It's big. So what we end up doing is we take a smaller subset of these molecules, right? We take a smaller subset of this molecule and say, I'm going to sample, about 1 million molecules and then choose the top molecules, right? So what will happen? You will end up identifying these red ones and then you will completely miss out on the green ones because I'm not even sampling this area. So the first question that we are asking is, how do I identify all these green dots and the red dots by doing minimum number of docking calculations, right? That's the first question we're going to tackle. The second question that we are going to tackle is related to the overall chemical space. What does it mean? The overall chemical space, or one of the estimates of how many molecules can you come up with which are drug-like, right? The estimate is about 10 power 60, which is impossible to sample, right? So the second question that we are asking is, can we explore this region efficiently, right? So that's what we're saying. So by sampling only a smaller space here, the first is, can I enhance the sampling to this whole region? And then the second question is we're asking is, by sampling this region, can I explore the unknown chemical space? So that's the question that we're asking. So in that context, the first method for the enhancing, we have something called a MEMS, which is the machine learning framework for enhanced molecular screening, right? So I will try and explain what it is. If you have any questions, let me know. So this is the flow chart of the method, right? What is the objective? The objective is this, where I have a ligand library, let's say the zinc library. Okay, I have a target. Let's have a protein. right? I want to rank all the molecules in this with respect to the docking results here and then come up with the top hits. That's my objective, right? So, but the problem is this library is so huge. I am not able to do docking on all of them. Okay. So now I want to do it more efficient. That's where all the other components come. So initially, we did this experiment up to 100 million molecules. Okay. So what we do is we represent these ligands in a certain way. We will not go into the details of this, but the idea is that the molecules in this ligand library are represented in a latent space, and we are working in that latent space now. Okay, so that's basically the molecular representation. So now we take this and do clustering in this space, and then choose representative molecules from each cluster, and that's basically this. So the number of molecules here now is a very, very small subset of all of the molecules present here. That's all we have done so far, right? So let us assume that, I have about 100 million here. Maybe I have 10,000 molecules here, okay? I do docking on these 10,000 molecules, okay? I start from 100 million. I reduce it to 10,000 here. I do docking on these 10,000 molecules, and I have the docking results. So using those results, I train a machine learning algorithm. So in this case, we use what is called the Gaussian process. We train the machine learning algorithm. Because I'm using very, very less number of points here, this is not going to be a very accurate model, right? But the objective is not to get an accurate model. My objective is to get as many top molecules as possible by sampling a smaller subset in this one, right? So that's all I'm interested in. So once you have a model, the advantage or the specialty of this method is that it, in addition to giving a prediction, let's say y as a function of x, in addition to giving us value of y with respect to any given x, it also gives us the uncertainty, right? Or the confidence of the model in predicting the output for a given input, right? We use that information and pick more points from the original data set and then redo the docking on those models and then retrain this model. And then we do this iteration, and once we exhaust all our computational time, you return all the top hit molecules, right? So maybe I think the next slide will become, the results will give us more insights. So let's look at this graph, yeah? So let's concentrate on this red line here, okay? The red line. So what is plotted? So this is the percentage of molecules or a fraction of molecules that have been identified as top hits, okay? So for example, let us assume we have 1,000 specific molecules which are your top hits, actual, okay? Out of this 1,000, how many do you actually identify, or how many do you capture? Okay, if I sample 6% of the data set, okay, if there are 1 million compounds, I am sampling 60,000 molecules. If I sample 6%, my fraction is close to 6%. So what this means is that, with respect to increase in this x-axis number, okay, this will also increase approximately linearly and then meet a value of 1 at a value of 100 here, which is expected. But then if you use our method, which is means, right, just by sampling 6%, okay, the maximum that you have achieved is about 96 or 97% in terms of identifying all of the molecules. So that's basically what we mean by, we invest less and gain maximum returns, right? So, there are other variations to this that we tried out, and they work very, very well in terms of identifying molecules or making the virtual screening experiments or high throughput experiments very, very efficient, right? So this kind of gives the overall picture. So let us say this is my whole data set. Yeah. So I'm taking the whole data set, doing docking on all of the molecules, 100% of the molecules, and I identify the 500 molecules. That's the ground truth now. If I pick random 6% randomly, I identify only 34 out of 500. That's very, very small number. But if I use memes to sample the same 6%, right, you can see that we have identified 490 or 489 molecules out of 500, right? So that's a huge advantage. By doing exactly same number of calculations, we are achieving very high number here, right? So now we can expand this to multiple properties. What do you mean multiple properties? Initially, we worried only about the docking. So now you can add more properties and then say, I want to, screen molecules with respect to multiple properties. You can do the same thing. You can extend it to this and then start identifying molecules that are of your interest, or maybe for more focused search of the molecules. And the second problem now we want to explore, right? What we spoke about is enhancing by sampling a smaller subset. How do I, explore all of the known chemical space? The problem now that we are going to talk about is by sampling the same smaller subset, can I explore a larger chemical space, which is unknown? So there are several methods that, one can use, right? One of the earliest one is a genetic algorithm method that was proposed, and then they proposed RNN, or the recurrent neural network based methods, or GAN, or the variational autoencoder, reinforcement learning, and so on. So let's talk about the last one today. So we have used multiple of these methods for this inverse molecular design, but we'll talk about this today. Yeah. So let us say my interest, what is the interest? My interest is I want to come up with an algorithm which will generate new molecules that are not there in the data set, not any random molecules, but molecules that exhibit certain properties, right? So what do we have here? Let's go one by one. So the top one is a molecular generator, or a network which can generate valid molecules, right? This is not so complicated task given the amount of data that we have on molecular organic molecules, right? So you can, develop RNN or any language-based models to come up with this network which can generate new molecules. So let us assume I have this generator which gives me a molecule, and then I take the molecule and predict a certain property that I'm interested in. If the value of the property is close to my requirement, then I reward the generator saying, oh, you have done well, so I'm going to give you a reward. On the other hand, if the property value of the molecule that is generated here does not meet my requirement, then we penalize this generator saying, you did not generate a molecule that is of my interest. You generated a molecule which I have no interest in. So you penalize the molecule. Okay. So now you train this whole network, a whole framework, and by maximizing the reward, right? Slowly the generator will start to produce molecules that have certain set of properties. Let's take an example, right? So I'm looking at two properties here. One is the log-P. Again, most of you may know the log-P value. Just for a quick recap, log-P values are related to the permeability of molecules across cell membranes, right? If the log-P value is between 0 to 5, then it is considered that the molecules can permeate the cell more easily than when the value is less than 0 or greater than 5. Okay. So what is the objective? The objective is I want to generate molecules that have values between 0 and 5. Second is synthesizability. So there's something called a synthetic accessibility score, which tells us how difficult or how easy it is for you to synthesize a given molecule, right? So again, this is an empirical score, which, higher the value, or this ranges from 1 to 10. If the value is close to 1, that means it's easy to synthesize. If it is close to 10, it's more difficult or much more difficult to synthesize. So here, what is the objective? I want to generate molecules which are easy to synthesize, right? So if going back here, what are my interests? One is the log P should be between 0 and 5. And second one is that the synthetic accessibility score should be close to 1 or towards 1. That's my objective. Okay, so that's what we have here. If I don't have any objective, or if I don't train the model without, or if I do a random generation, right? So this is how the distribution looks like with respect to the log P values, right? So that means the value ranges from approximately -3 to all the way up to 7 or 7.5, right? So, but what is my objective? Objective is for me to generate molecules in this range. Why? Because those are the molecules which have higher chances of permeating across the lipid bilayer, right? I'm not interested in molecules in this region or in this region because they cannot permeate. In this case, it's a synthetic accessibility score. If I don't have any condition, this is how the distribution looks like. In this case, I don't want molecules, which are very, very high in terms of the synthetic accessibility score, but I want closer to here. Now I start the training, and you can see with respect to the reward system, the molecules that are being generated now slowly move towards these two windows. Right now, compared to before, which is in gray, now almost all the molecules are having values of log P between 0 and 5. Similarly here, with respect to training, the value of the synthetic accessibility score of the molecules that are being generated slowly comes down, right? So now you can use this method to, make the objectives more complicated. For example, in this case, I want the docking scores or the binding affinities to be as favorable as possible, and then the log P value to be favorable for permeation. I use the same method. I have a generator. I have these objectives, which is the docking score, and then the log P value, and then I use the reward system or the actor critic policy gradient method to generate new molecules. And we can generate new molecules which are not there in the data set, and which meets our objectives. So that's how we generate new molecules. Yeah. So and then this next one example is this is you must have heard about ChatGPT or GPT-3, GPT-4, and so on. It's in exactly the same method, right? Where you use the transformer decoder model to generate new molecules. These are language models, right? You generate new molecules with respect to certain conditions. Yeah. So, for example, in this case, I want molecules which have these values, and then you see this. The red distribution is the initial data set values, and 2, 3, 4. These three are the ones which are generated molecules, right? Again, you can increase the number of properties to 2 or 3 or 4. Like, for example, here you can also condition it based on the existence of certain scaffold. Like, for example, I want to generate molecules with all of them having this scaffold, right? You'll see that these are all different molecules, but all of them have this particular fragment of the skeleton, this particular moiety in all of them. Okay, so the last one I wanted to quickly show is spectra to structure, right? So if someone gives us structure, getting or modeling the spectrum is not so difficult, right? There are programs which can produce very good C13 or proton H1 NMR, right? Very efficient. But then that's not the problem we want to solve. We want to give the spectrum to a software, and that software should tell us what the structure is. That's basically what we want. So here again, we use a type of reinforcement learning method, which is called the Monte Carlo research method, where we go from spectra to structure, right? So it's, I'm not going to the details, but the idea is this, where you have the spectrum and the molecular formula that as an input to the network, and then slowly it builds the molecule, right? And then, comes to the target molecule, which matches the NMR spectrum, right? As you can see here, it starts from a methane molecule, right? And it ends up with this particular molecule, which is actually the molecule to which this particular spectrum belong to, right? And these are the accuracy and so on in terms of how well the algorithm does, right? So to quickly summarize, I think the first half and this one, I tried to show some examples of problems and how it can be used to solve these problems using machine learning, right? So, and you can see that, we are able to solve a diverse class of methods, and it is, the machine learning methods in general are, proving to be a, good addition to the computational toolkit that we already have. And then, some examples that I showed are, efficient sampling of chemical space, where we get the approximate function and the uncertainty in the prediction to make the virtual screening exercises much more efficient. And then the other one I try to show is to do inverse design, where I say that I specify the properties and the algorithm gives me molecules, right? And also I showed a quick example of how you can start from a spectrum and then get the structure from these values, right? So with that, I will stop, and if you have any questions, you can ask, or yeah, please go ahead.
Thank you, sir. Thank you very much for your informative session.
Should I unshare my screen?
Yes, sir.
Yeah, anyway, maybe before I finish, right, I mean, we have a number of these methods developed, and all of these methods are also available in our lab, GitHub page. So if someone wants to use, or, please do feel free to visit the GitHub page and then see if we can use it. Yeah, I'll stop sharing my screen. Thank you very much.
So I request our organizing secretary, Dr. K. Rajalakshmi, to address the gathering.
Thank you, Shailu. I feel it a great privilege to extend vote of thanks on this occasion. First and foremost, I thank Lord God Almighty for strengthening us in organizing this 1 week online faculty development program. I express my profound gratitude to our chairman, sir, Dr. Lau Rataya Garu, Vice Chairman, Sri Lau Sri Krishna Devarailu, and Principal Dr. P. Srinivas Babu sir for their support and encouragement in organizing 1 week online faculty development program on emerging trends in drug design and development. I extend my thanks to all the resource persons for their exemplary training, first aid Talk delivered by the Professor Prasad V. Bharatam Garu, Professor and HOD of Medicinal Chemistry, NIPER. He delivered lecture on drug design, PPAR gamma agonist design and AI drug design. Second day, talk delivered by Devalapalli P. Ramachari Garu, Professor of Chemistry, School of Chemistry, University of Hyderabad. He delivered talk on discovery of parts per billion level catalyst asymmetric cannulations. Third day, held with Dr. B.M. Gurupadaya Garu, Professor HOD of Pharmaceutical Chemistry, College of Pharmacy, JSS Academy of Higher Education and Research, Mysore. He delivered lecture on bioanalytical approaches in drug discovery and development. Fourth day held with Dr. V.M.K. Khathiravan sir, Professor and HOD, Department of Pharmaceutical Chemistry, SRM College of Pharmacy, Chennai. He delivered talk on drug design, a medicinal chemist approach. And next, third day session was held with two eminent resource persons. That session 1 was delivered by Dr. K. Suresh Babu Garu, Senior Principal Scientist, Department of Natural Products and Medicinal Chemistry from Indian Institute of Chemical Technology, Hyderabad. He delivered lecture on contribution of natural products from for human well-being. On that day, second session was held with Professor Pullo K. Mukherjee sir, Director, IBSD, Department of Biotechnology, Ministry of Science and Technology, Government of India. He delivered talk on ethnopharmacology and bioeconomy, developing drugs from nature and traditional medicine. So today's session was held with Professor Devapriya Kumar Garu, Professor and Head at International Institute of Information Technology, Gachibowli, Hyderabad. He delivered talk on modern artificial intelligence methods for drug design. I thank all the participants making this online 1 week faculty development program a great success, and my special thanks to supporting staff and technical support. Once again, thank you.
We will close the meeting with national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता। पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा। विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा। तव शुभ नामे जागे तव शुभ आशीष मागे। गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता। जय हे जय हे जय हे जय 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 हे।